welcome to the n-Lorem podcast series, a series that focuses exclusively on patients now referred to as having nanorare mutations. I'm Stan Crook, and I'm the founder, chairman, and CEO of n-Lorem. n-Lorem is a nonprofit foundation that I initiated in January of 2020. Our mission at n-Lorem is to take advantage of the technology we created at Ionis Pharmaceuticals, Antisense technology, or ASO technology, to discover, develop, and provide experimental ASO treatments to nanorare patients, and to do that for free for life. And today we're going to continue our conversation about sort of graduate course in genomics. And for those of you who survived the last lecture, welcome back. In our last conversation, I began with the basics of genetics and your cells used to store, retrieve, and use genetic information to lay the groundwork to help you understand how we look at various types of genetic mutations to make judgments about whether a mutation expressed in a patient is amenable to treatment with an ASO. If you missed our first episode on genetics, I encourage you to listen to that one first and then join us for this one. The question that I left at the end of our first conversation was, why are some mutations pathogenic? That is, why do some mutations cause disease and others, so many others, do not? Let's talk about the easiest mutations to understand first, SNPs. Remember that a SNP is just short for single nucleotide polymorphism, which is just a complicated way of you have a mutation that causes a change in one genetic letter only in your genome. Now, single nucleotide changes that don't change the reading frame. Remember, we talked about the messenger RNA is read left to right, and there are signals to start translation and signals to stop translation. So when we talk about reading frame, talking about the part of the message that begins with the start signal, AUG, and ends with a termination signal that says, stop translating me. I no longer am making a protein. If you have a mutation that doesn't change the number of nucleotides, the reading frame will be the same. And so if you have a mutation, for example, instead of UUU, you have a UUC, and that codon is redundant, and you still put a phenylalanine in, then you haven't changed anything in the protein. The protein is still the same length, and it has phenylalanine where it's supposed to have it. So there are some SNPs that are silent because they don't change the reading frame, and the genetic code is redundant for some amino acids, and a different set of letters can give you the same amino acid. Alternatively, some amino acids are chemically similar and can substitute for one another. So a codon change that caused, say, serine to be put in a protein rather than leucine may not make a protein that's any different at all. And so sometimes you have what's called isosteric amino acid replacement. You have a change in your letter that results in a change in amino acid, but an amino acid that's chemically similar enough that you make a protein that functions as the good protein would. The final way, as long as the reading frame isn't preserved, that you can have a mutation that's silent, is that you have a mutation in a part of the protein that doesn't matter. And so proteins are big. They have a lot of different things in them, but not all of the things that they have in them really matter. And so if you had a mutation that caused a different amino acid to be placed in a part of the protein that really isn't essential for function, then you could have a silent mutation. That is at least one means by which you can have mutations that are silent, that is, that don't produce what we see as disease. Now, the SNPs that do cause problems come in several varieties. They may cause a change in amino acid that's critical to the function of a protein. They might end up making a protein that has the wrong activity, is inactive, or actually even has greater activity and that can do you harm. A mutation that causes a loss of activity is called a loss of function mutation, A mutation that causes a significant increase in activity or a change in activity is called a toxic gain-of-function mutation. And sometimes these mutations in proteins can make a protein that will block the function of the normal protein. And that's called a dominant negative mutation. So you have at least three forms of disease-causing mutations. Mutations that occur in amino acids that are essential for the protein and change its either increase its activity, decrease its activity, or alter its activity. Sometimes, and acid shifts that can cause the mutant protein to actually interfere with the function of the normal mutation. There are other kinds of mutations that can cause the disease too. Unfortunately, get even more complex. Remember that no mRNA, or almost no mRNA, starts out as mRNA. They start out as much larger RNAs called pre-mRNA or premature mRNAs. And in the nucleus of your cell, those RNAs have to be processed, trimmed to make the final mature messenger RNA. And I hope that you recall that the pre-mRNA have extra segments of this genetic code that have to be excised. And then the RNA reconnected or respliced. And so that whole process is called RNA splicing. And so you can have mutations that affect any step in the process from transcription to shipping the mature RNA out, mRNA out to the cytoplasm to make a protein. So SNPs can result in several types of consequences. No effect. There are many silent mutations, which means that we don't really change the function of the protein in a meaningful way. And of course, you then don't have any change in your phenotype that you perceive as a disease. Loss of function mutations can be the loss of the entire protein that happens, or it can be loss of function of a protein. That is, an inactive protein is made. Toxic gain-of-function mutations cause the protein to do more of what it's designed to do, or even something that it's not designed to do. Either way, it can be bad for the cell. And then dominant negative mutations are mutations in the protein that actually make a protein that prevents the good protein that might be in your cell from doing its job. Sometimes you have mutations that create stop signals in the open reading frames. And these, again, produce truncated proteins. Sometimes the truncated protein is not active. Okay, it's a loss of function mutation. Sometimes the truncated protein is toxic. All of those things can occur in the mature mRNA. And then there's a slew of mutations that can happen in the pre-mRNA that alter the production of the final messenger RNA and then lead to disease. So yes, you have hundreds of millions of different SNPs. Most of them are silent. They may increase your tendency toward, say, having diabetes, or may increase your tendency toward hypertension, but they are not associated directly with 100% certain production of a disease. And the vast majority of them may be good. They may make you run faster, or they may, but they're all mutations. Now, indels are, of course, more problematic because you've had insertion or deletion of a big hunk of genetic information. What happens with indels will depend on how long they are. Do they fit in with 3 nucleotides in a row in the same register as the normal message? In that case, all that you've done is you've inserted a set of nucleotides that will be read and put into a protein. And now it'll make a bigger protein. Or suppose you have an indel that deletes a specific number of nucleotides. As long as it's still the unit of 3, you'll make another protein and it will be shorter and may have different activities. On the other hand, an indel can be some odd number other than 3 times 3 times 3, in which case it would change the reading frame. It might even produce a totally different kind of protein. So indels can occur anywhere in the genetic code, and they can lead to all kinds of problems. Indels, because they're very large hunks of DNA that are gone or added or whatnot, are, of course, much harder to deal with than SNPs. So indels can disrupt reading frames. That could end translation, resulting in a truncated protein being made, or be in the reading frame and produce an even larger or smaller protein that would have a different function or be toxic. And so indels are much more likely to produce disease than SNPs, and they're much more challenging from a therapeutic point of view to deal with. Let's just define a few terms. Let's begin with allele. All cells in your body except sperm and ova have 2 copies of every gene. And 1 copy is from your mother, 1 copy is from your father. Those individual copies are called alleles. So you have a maternal allele and you have a paternal allele. So an allele just means a copy of a gene. Then homozygous. What does homozygous mean? It means that you've inherited a mutation, the same mutation from each parent. This can happen. It just happens all the time. Hemophilia, for example, is an example of just the bad luck of 2 parents with the same mutation getting together and producing a child who has the disease when neither of the parents had the disease. These kinds of homozygous mutations occur in both alleles. You get a mutation from your father, you get a mutation from your mother, they're the same, you're a homozygous gain or loss of function mutation. And as you might expect, the most common sorts of places where you see these are in populations that have been significantly intermarried and inbred for a long period of time, like the Amish or some Jewish populations. As a general rule, a homozygous mutation is typically more severe than a heterozygous, which brings us to heterozygous. Most of us inherit a mutation from one parent and a normal gene from the other parent. In that case, you're a mosaic. You're making a normal protein, you're making an abnormal protein, and you're called a heterozygote. And that means that all the cells that have the 2 different genes are mosaics. They're making 2 different messengers, they're making 2 different proteins. And all things being equal then, if you have 1 copy of good protein, most of the time that's enough. So heterozygous mutations tend to cause less severe disease, and heterozygous mutations are things that we can deal with much more easily than homozygous. And then there are things called, because everything has to be more complex, compound heterozygotes. And that is, you get a mutation from your father that affects a particular gene that makes a particular protein, and another mutation that affects the same protein from your mother. Now you have 2 different mutations in the same protein with both alleles affected. And so that can be called a compound heterozygous mutation. So you have 2 genes per cell. Mutations can occur in both of those. If you inherit 2 mutated genes, one from your mother, one from your father, you're homozygous for that trait. And typically, that's going to be a worse disease than if you're heterozygous. If you're heterozygous, you've got 1 bad gene, 1 good gene. And if you're compound heterozygous, you're really complex. What about genomic sequencing? Well, the first step in identifying whether a patient has a genetic disease is to sequence his genome. And there are 2 levels of genomic sequencing. Some you've heard of as whole exome and some whole genome. Whole exome refers to sequencing the coding regions of genes. And whole genome sequences the whole genome, all of the information in the genome. And obviously, whole genome sequencing gives us a lot more information, will help us identify mutations that don't occur in the coding region that affect the performance of the message in the protein. And so as a general rule, we much prefer whole genome sequencing to whole exome. Whole exome is done because it's less expensive and easier to do. But whole genome is really a much more inclusive and helpful approach. If you have a genetic sequencing, you can learn that you have a mutation, but as many patients of ours have learned, that's just the beginning. You haven't learned whether that mutation is causing a disease, you haven't learned the nature of the mutation, and you haven't then enough information for us to make a decision about whether we can treat you or not. Sometimes, if we've seen precisely the same mutation in another patient, we're going to know right away. that this is a disease-causing mutation, it has these characteristics, and we can go straight from there and make ASOs if it makes sense. And the more experience we have in the nanorare patient population, the more genes that we have sequenced, the more mutations we understand, the more likely it is that we'll be able to make these decisions even more rapidly and do better and get to patients more rapidly. Recognizing previous mutations that have been seen and characterized is a really important step in shortening the process. If we don't have any prior experience with the mutation, or if the experience is very limited, or there are questions, then we have to do a lot more work to find out, is that mutation actually causing the problem that the patient has? And what is the nature of that mutation? Genomic sequencing is necessary for us to understand our patients, but it's not sufficient. We have to have more information about the type of mutation and information that convinces us that that mutation is really causative. All of that takes a lot of work and a lot of time. Sometimes it's done in the academic centers. Sometimes we have to do it. In some cases, we may be able to move very rapidly, and other cases may take a long time before we really understand enough to know whether we can help a patient or not. Now, on to how do we decide? We get an application, and we've had now 250 or so applications, and we are able to say yes to about 110 so far. So not quite 50%. How do we make that first choice? Well, one of the important attributes of antisense technology is versatility. And because of the versatility, we can design ASOs to do different things in the cell. And therefore, there are more types of mutations that we can address than you might think. And we also know that we can give ASOs by all routes of administration. And we have a very good idea of the organs where we get good activity, relatively limited activity, where we're going to have a good chance for doing something good without side effects. The first step is that we look at the prevalence of the mutation. If it's within the nanorare definition, that's step one. The second step is to ask, where's the patient located? Right now, we're working in the US. We hope to be working in other countries soon. The third step is much more complex, and that is, what is the mutation? What is the nature of the mutation? And then what is the evidence that is causative? And then when we've looked at all of those things, we can make a decision about whether a patient is treatable. Now, there are some mutations that we can't address. And those are typically called null mutations, meaning you're not making a functional protein. Null mutations are simply where you're going to have a gene, and we can't help those patients. The patients that we exclude typically have mutations that we can't help, or disease in an organ that we're not comfortable treating, or they have a mutation that's much more prevalent than we are allowed to treat by the FDA, or they're located in some part of the world where we simply are unable to provide ASOs. We have to understand the mutation, the phenotype. We have to thoroughly understand the nature of the mutation, and then we can make a decision about whether we can design an ASO that might work. And then if it's in the right organ and in the right other characteristics, we can go to work. How do we design ASOs to take advantage of different post-RNA binding mechanisms? Well, that's just a product of all the decades of work that we've done. And the most common, the most useful mechanism for ASOs is called RNase H1. RNase H1 is just an enzyme that recognizes, remember I called it an ASO RNA heteroduplex, recognizes that and cleaves the RNA in that heteroduplex. And because RNA is cleaved, then it doesn't make the protein. So an RNase H1 ASO is always designed to treat a toxic gain-of-function mutation. And to do that, we design ASOs called gapmers. And the reason they have to be gapmers is we have modifications of the ASO that increase stability and potency and safety. But the center of the ASO has to look like DNA for the enzyme to work. And so those designs you'll hear a lot about, and they're called gapmers. The most common modification that we use to produce increased potency and tolerability and safety and duration of effect is 2'-methoxyethyl, or MO. And so you'll hear a lot of times we're talking about a MO gapmer. It's not complex, it's just, different types of chemistry that we use to build the best ASO we can. The RNase H1 mechanism is by far the best understood, most robust ASO mechanism, and that's always easiest. Many of our patients are heterozygous. That is, they have a good copy of a gene and a bad copy of a gene, and their cells are mosaic, making good RNA, good protein, bad protein. Many of those genes produce proteins that are essential for life. And those present a little more challenge for us because what we need to do is design an ASO that will only cause H1 to cleave the bad RNA. And to do that, what we have to find is these SNPs, these mutations that we've talked about, that are silent. So we know we have a pathogenic mutation that's causing the disease. We can't use that, but we then scan the rest of the RNA and find where there are single nucleotide changes that don't cause a change in function or disease. And then we can use those to design ASOs to differentiate between the wild type and the normal. That's a challenging task. We have to find those sites. Sometimes RNAs are very small and there just aren't any. And if we don't have a non-pathogenic single nucleotide change that we can use to differentiate between the two, we simply can't help. Sometimes we have many of them, but it's very hard to find a potent, safe, allele selective ASO. We're learning to do better at that. We have a research program now that I think is going to help us make much better allele selective ASOs in the future and do it more rapidly. So that's another mechanism we use. And then we can use ASOs that alter splicing. Spinraza is a splicing ASO. And so those can be used for diseases caused by mutations that disrupt the processing of pre-messenger RNA. In fact, there are many other mechanisms and designs that we can build in ASOs, but those three give you a sense of the versatility. And one of our goals at n-Lorem is to continue to advance technology, that there are more and more opportunities to design ASOs to do different things in the cell. So the key point here is that we have the versatility to design ASOs to take advantage of different types of mechanisms to address different kinds of genetic problems. There are some problems that we can't address, and some problems that are more difficult than others. Creating an allele selective ASO is intrinsically more difficult than creating a non-allele selective ASO. That brings us to the conclusions of these two way dense lectures. I've been trying to give you a much more detailed understanding of the nature of mutations and help you understand why some patients seem affected by a single nucleotide change and other patients seem unaffected, and why that happens, and that we are all an assembly of an incredible number of mistakes. Luckily, most of those mistakes do not produce disease. The second big lesson that I wanted to provide is all of the work that has to go into and all the information that we need to make an informed decision about whether a patient who's applied is appropriate for us to say yes to, so that we can try to make an ASO for that patient. And it's complex. So when you ask, why does it take time? Why does it take so much effort and so on? It's because biology is complex. Medicine is complex. And disease is enormously complex. And our objective is to provide ASOs that have a good chance of bringing benefit without producing severe or significant adverse events. And that takes time, it takes understanding, and it takes a great deal of effort on our part to do that. Now that you understand graduate level genomics, I hope that you have a better appreciation of all the challenges that we have to face as we determine whether we can help you or your loved one, and then as we work to do that.
n-Lorem is a nonprofit committed to discovering and providing personalized experimental treatments for free, for life, to patients with genetic diseases that affect 1 to 30 patients worldwide, referred to by n-Lorem as nano-rare. Many of these patients progress and die without ever achieving a diagnosis. This is where n-Lorem comes in. They do the impossible by providing hope, and for those that they can help, free lifetime treatment. For more information about n-Lorem or today's episode, visit n-Lorem.org. Any questions can be sent into podcast@n-Lorem.org. Search n-Lorem on Twitter, Instagram, YouTube, LinkedIn, and Facebook to connect with us. Please rate and review the podcast on Apple, Spotify, or wherever you listen. This truly helps us climb the charts and allows others to find the show. This podcast is hosted by Dr. Stan Crook. Our videographer is John Magnuson of Mighty One Productions. Our producers are John Magnuson and Kira Deneen of DNA Today. Thank you for listening.